All right, so let's talk about renal physiology now. So just a quick review, this all is based upon osmosis and osmolarity, the hydrostatic pressure, and so on. So just remember as a quick review of that, what is osmosis, right? That is simply when the concentration of solutes is, say, high on one side of a semipermeable membrane, and then water is going to go from the concentrated side, the lower concentrated side, to the high concentrated side in order to equalize that, right? For example, we could say if you have this, let's say you have a container of water right here, and you put a semi-permeable membrane right in half, right there, and now you filled it with water, right? This is just to put water in. Well, the water's gonna be able to go through both sides, and you're gonna see that it's gonna level out, right? It'll be level. And now, if you were to put a whole bunch of salt into one side of this, right? So here's a bunch of salt, right? And here's your salt shaker you're doing, right? You're putting salt in there. So now there's a whole bunch of salt in the water. What's going to happen to the water level, right? Well, now you're going to have a new water level. And the new water level is going to look like something like this. This is going to be up here higher, and this is going to be down lower, right? And the reason for that is, again, it's because you have all these solutes over here. These are osmoles, right? And the water is going to come through that semipermeable membrane, and it's going to fill that side, and that's going to make the water levels unequal. Well, why is it that the water doesn't all go over there? Well, it's because of what we call the hydrostatic pressure, where that's the water on top, right? All this water up here is pushing down, trying to force the water through that membrane, right? That's kind of how reverse osmosis systems work as well. It's pushing the water through that membrane, and you get filtration. In this case, you have this osmotic gradient that you've created. So you have two forces that are directly opposing each other, right? Hydrostatic forces and the osmotic forces. And as another example you could think about is a balloon, right? So if we take a balloon and we have it, like, let's say we have a balloon here, and then we're going to put some holes in the balloon, all right? Just really, really small, like pinholes. And inside the balloon is full of water, and you have like large molecules, proteins, let's say. And now the water, of course, if you put those holes in there, the water is going to want to come out. Why is it going to want to come out of those holes? Because the hydrostatic pressure is forcing that water out. But in this scenario, not all the water will go out, because those proteins that cannot go through, they're pulling the water back in, right? That's the osmotic pressure, or the colloid osmotic pressure. And when we talk about proteins, that's specifically the oncotic pressure. That's the colloid osmotic pressure. Whereas just ions, that's the just normal osmotic pressure, okay? And this is a very important principle to understand in order to understand renal physiology and what's happening here in the kidney, okay? This is specifically the nephron. Now, the kidney is, as you know, is responsible for like blood pressure, filtering the blood, and a number of other things that are so important. But it's amazing that each kidney has a whole bunch of these guys right here called the nephron, and there's like 800,000 to 1.2 million, so around 1 million nephrons per kidney. And these nephrons are responsible for filtering the blood. And here we're drawing a juxtamedullary juxtamedullary nephron. And what that means is this is the type of nephron that goes really deep down into the medulla. So if you think about the anatomy of the kidney, you have the cortex. Okay, so I have labeled here, like this is the top part where the cortex is, and a lot of the nephrons and the loop and everything, they're kind of just up here. But some of them, around 20% or so, are going to go all the way down into the medulla, and that's where we can get a very high concentration of solutes and how we can conserve water. So this has a lot of implications when it comes to our hydration status, or what's going on with dehydration, conserving sodium, our ions, and just filtering our blood. So we're going to walk through all of that, but just so you're oriented. This is what I drew as a nephron. Now, in your text, you'll also see that the nephron is not really like this, like I kind of opened it out, but actually the nephron's kind of folded in on of itself. And so I have like this over here. This is referred to as a macula densa. And this part is actually right here. So they're like right up next to each other. So that's going to be important for some of the sensing that we're going to talk about. And then the other important part is we have the efferent arterial and the afferent arterial. So this is where blood is coming through the afferent arterial, coming into the Bowman's capsule, right? This is the juxtaglomerular apparatus right here. And this is where the filtration is going to happen. And then the blood's going to come out to the efferent arterial and come around this way. So as we're drawing it, yes, blood's going this way. But remember, if this is folded off, then actually the blood's going to be going in the opposite direction that the filtrate is going. Okay, so it'd be the opposite away. So just keep that in mind, because when we talk about the countercurrent multiplier effect, that's what's actually going on. All right. So now let's get into this. Let's go into some of these the anatomy a little bit, so we're oriented. Okay, so this, as I said, is like the Bowman's capsule. Here, this is the glomerulus, okay, or the juxtaglomerular apparatus. Right here is a tubule. It's close to over here, and so we would call this the proximal convoluted tubule, which we can just put proximal convoluted tubule. And then as we come down through here, this is called the descending limb of the loop of Henle, basically. So we can just I'll just put DL for the descending limb. And then here is the loop of Henle. I just would put L, well, yeah. Loop of Henle for LOH, that's what that can stand for. And then here's the ascending limb of the loop of Henle. Okay, so ascending limb, just AL for ascending limb, loop of Henle, and this is the thick portion of, the ascending limb of the loop of Henle. And then it turns into this part. This one's further away from over here, so instead of proximal, it's going to be called the distal convoluted tubule. Okay, so we can just put distal convoluted tubule. And then over here, we have the collecting duct. Okay, this is the, well, I'll put just CD for collecting duct. And this is where all the waste is going to go. Because once it comes out here, this is going to, this over here is what's going to be excreted. Okay, so this is excreted. In other words, this is what is going to make your urine. All right. And all these mesh together so they all connect to this collecting duct, and then it's excreted into the urine. So let's go through some of the things that are happening here. First, as I have I drew the efferent arterial. Remember, the blood's coming this way, and it's actually interesting. This is one of the few times where you have an arterial itself going in and then coming out, and it's still an arterial. So afferent arterial, efferent arterial. So as it comes through, these here are called the peritubular capillaries when it's up here more in the cortex. Now, when it comes, these capillaries, when they come down here, they kind of get modified, and we would actually call, and I kind of wrote it here just so we can orient us a little bit. But this has a different name, but this is called the vasa recta. Okay, and this is going to be important for absorbing the salts and doing the countercurrent multiplier effect that we talked about. So the whole process here is to do filtration. And in order to do filtration, we need to get this blood to be filtered through here, right? And now remember, blood it comprises your plasma and then the formed elements. So in your plasma is, right, just mainly water, right? And your formed elements, that's going to be like all your cells. And so if you have 5 liters of blood or so, then around 2.5 of that, 3 liters is going to be plasma, and the rest is going to be the formed elements. So in order to look at the filtration process, it's going to be based upon these pressures, these osmotic and hydrostatic pressures. And so there's an equation actually we could look at with this. So if we take our pressure, all right, we would say our pressure of the hydrostatic pressure, right? So we can say P, this is a little h, not to get confused with pH, the potential of hydrogen ion, right? This is pressure. And if we take the hydrostatic pressure and we're going to subtract that from the osmotic pressure, the colloid osmotic pressure, okay, and that's going to be represented by here. And then we can subtract that by the pressure of the fluid of the capillaries, or inside of right here, okay? And then that right there is going to equal our net filtration. Okay, so we get our net filtration. And we actually know what these numbers can be measured. So if we look at how much of the pressure, of the hydrostatic pressure is pushing the blood through this filtration, well, on here, it's going to be around 55. So we get 55 torrs a milliliter of mercury here. And then we're going to subtract that from the oncotic pressure, or the colloid osmotic pressure. That is the amount of proteins that exist inside the blood, inside the plasma, okay? And when you do and that is equivalent to around 30. So that's the colloid osmotic or oncotic pressure. And then you need to subtract the pressure of the fluid, the pressure that's inside, like here in the lumen, the Bowman's capsule, that's pushing the fluid back inside. It's not going to be very much, right? So that's going to be around 10 or so, all right? So we subtract that, and then that's going to equal our net filtration, right? So 30, oh, that's right, that's going to be 15. Did that wrong. And we subtract that from the fluid that's going to be inside the proximal convoluted tubule, right? That's not in the Bowman's capsule, but you're also having some of that pressure going into the capillaries, let's say, into the plasma, and this is around 15. And so when you do that math, you end up getting around 10 millimeters of mercury. So you have a positive net filtration. So this is where the net filtration is coming from. So the blood is literally pushing things through there. And in these, there's different sites like podocytes and a number of things that happen, and these capillaries that are helping. And there's some important things about this, because this has like there's fenestrations. Okay, I'll just put fenestrations. And they're just really small holes in these capillaries that are going to allow only small molecules to get through. And so the hydrostatic pressure is pushing the entire blood through there. You only want to filter out the liquid things. You don't want to get the big proteins. And so you have a number of barriers that are going to prevent that from happening, such as the fenestrated capillaries. You also have like glycosaminoglycans and heparin sulfates and other things that are going to repel other negatively charged proteins like albumin and so on. And so in the end, what is coming out of here, you're filtering, all of your small molecules. So your glucose, your amino acids, all of your ions, you have water. All of these things are going to be coming through the filtration, whereas your bigger things like albumin and other immunoglobulins and so on, they're going to stay in the plasma, and that's going to result in this oncotic pressure. Okay, let's pull it that way. So that's the first stage, is the filtration. All right. Okay, I just want to remove that equation up here so you can see me better. Now we're talking about filtering this blood, and in fact we have like, around let's say 80% of the blood is going to be coming through this area right here. Well, how much is going to be getting filtered actually? Well, it's around 20%. So you get 20% here, and then you end up reabsorbing back into the blood, right? You have all the peritubular capillaries. You end up getting most of that is going to be reabsorbed, as we'll talk about. So let's first go through each of these steps. So you get 20% here, and then most of this is going to actually be reabsorbed right here into the peritubular capillaries. So we could say 19%, probably even more than that, will be actually reabsorbed. So a lot of that is happening just right here. Now, a lot of blood is going through here, and so we can actually calculate what that would be, it can be measured, and it's around 125 milliliters per minute. That's the GFR, the glomerular filtration rate, which equates to around 180 liters per day, which is impressive. Think about, again, the kidney is literally filtering 180 liters of blood every single day. And that's why it's important that you're reabsorbing almost all of it. So you're filtering, all the blood multiple times, and then because you're only going to be excreting out 1%. So just a couple terminology. When the blood comes through here, this is called the filtration process. All right. Now, there's another thing called secretion, and you actually can get molecules secreted into right here, depending on what they are, like different organic molecules or whatever from the blood that can't make it into here. This would be more of an active situation, but you can do secretion, and this is called secretion. Now, once it's in here, if it doesn't get reabsorbed, once it goes through here or gets secreted into the lumen of the nephron, once it's there, it's going to be excreted into your urine unless it gets reabsorbed. So this is how the kidneys work, is they take everything out and then they take back what they need. And you kind of think about like if you have a messy dresser door or something, right? You could go through and pick out just the things that you want or something, but it could take a long time, versus you could just dump everything out and then so now everything is like clean, and then just get the things back that you want. And I guess you can think of it that way. So in this case, you are, as I said, you're filtering out the things that over here are staying will be like your red blood cells, your proteins, all of your cells, your larger molecules, but you're filtering and you're getting your glucose, all your sodium chloride and everything. In fact, you are absorbing back right here around 67% of your H2O, of your sodium chloride, of your, let's say about 90% or so of your bicarbonate ions, and 100% under normal situations of your glucose and amino acids. All right, so again, you're absorbing out most everything. And this is all isoosmotic. Okay, so it's isoosmotic. And that means your blood is around 300 milliosmoles, and so around here you're also around 300 milliosmoles. Okay, and again, an osmol is simply if you have a mole of substance, then and it's a solute, then that's also going to be 1 osmol. Now, if you have something like sodium chloride, right? If you have like a mole of sodium chloride and that dissolves into water, then you have 1 mole of sodium and 1 mole of chloride. And so now you have 2 osmoles, right? So 1 mole of sodium chloride could result in 2 millimoles of osmolytes, we would say. Okay, so that's an important when it comes to maintaining this osmolarity, because again, we talked about over here how that osmolarity is so critical when it comes to doing like filtration or absorption. So everything's going to follow these concentration gradients. And that's really what we're going to establish right now, is what is this pathway that's going to create different concentration gradients so we can reabsorb various electrolytes and concentrate the urine during times of, say, dehydration, where we want to conserve that water. All right. And the way that's going to happen is this water is going to come through here. So it's around 300 milliosmoles. And this descending limb it's permeable to water. So we have water in here and it can just come right out, no problem. All right. So, and it's going to just follow its osmotic gradient. Okay. So water is coming out. However, ions, okay, salt, they're going to be, whether they're out here, sodium ions, or in here, okay, they are not permeable. Okay, they can't go through here. So this, boom, they can't get through. So this this is called the descending limb of the loop of Henle, basically, and only water is able to go through, whereas the ions are not able to go through. And it's going to come down, and the concentration gradient is going to start changing. And you're going to see why, but let's say we get down over here, and we're, say, 400 milliosmoles, right? And then over here, say 600. And it's going to get more and more concentrated. And you'll see why here in a little bit, 800, you could say. And then maybe even down over here at the very bottom, it could be 1200, all right? But it's also going to be 1200 out here, okay? And same thing, it's going to be 800 out here. So everything's going to follow its concentration gradient. And then as we come up, so now we're back over here to say 600. And then we can say 400. And then once we get over here, we could maybe say 200. And then maybe even like 100 over here. And now, how does that happen? What is going on to allow us to make such major changes in the concentration gradient? And there's two things that I want to focus on. First is with the descending limb, it's going down, descending limb. Again, it's only permeable to water, but it's not permeable to the ions. Now as we go down deep into the medulla, where the concentration can be reached as low as 1,200, even 1,400 milliosmoles, you also have a lot of, I just want to add, you'll have a lot of urea out here also, is actually a strong electrolyte. So how is this concentration gradient? Well, it's all about right here. Okay, so first off, you're going to have some ion channels in the thin portion of the ascending limb of the loop of Henle. And these are passive channels that are going to allow your ions to go through, so such as sodium. Okay, so you have like sodium here, chloride, of course, right? So these are just going to follow its concentration gradients, because as you move up, this is going to be very concentrated, it's 1,200 milliosmoles, and as it starts coming up higher and higher, the ions are going to want to come out. But what can't go is water. All right, so we'll have the opposite thing going on over here. So water can't go this way, and it cannot go this way. All right, so water simply can't go out either way. All right, and that's going to be very important because we want to. This is how we're making the concentration gradient. Once we get to the thick ascending limb, that's where we're really going to start making a very strong concentrations. We should probably put this one as 300 because we're at the thick ascending limb now, so we're really going to be fighting against our concentration gradients. And this is where we're going to have various active transporters that are going to be pumping out sodium and chloride. And I can draw this, let's see. I'm going to draw this a kind of a bigger cell, basically, because we want to show what's going on. Okay, so you have over here, you have a higher concentration of ions, and you're going to just allow through a transporter, you're going to allow your sodium ions to come in, and a potassium ion, and a chloride ion. Okay, so this is a sodium, potassium, 2 chloride. So it's 2 chlorides, okay? So sodium, potassium, and 2 chloride ions are going to be coming through this transporter. And then on this side right here, you have your typical sodium, potassium, ATPase that's going on, right? So you potassium in and your sodium out, okay? This is sodium, potassium, ATPase. I'll just put ATPase. And now and then the potassium, actually, there's another channel here where potassium can come back and forth to reach equilibrium. And this is going to be important, actually, because you can absorb interstitially or paracellularly some other dications, such as your calcium and magnesium. Calcium and magnesium. Okay, so let's go through this again. We have this portion right here is permeable to water. So there's lots of these aquaporin channels. I'll just put AQP, I think it's aquaporin 1 channels, all right? The aquaporin 1 channels are going to allow the water to go through, and it wants to go through on both sides, of course. This is going everywhere. It wants to go through because you have all of these ions that are getting pumped out from the ascending limb of the loop of Henle in the thick portion, right? You can put in all these ions out there. Now, as you come through, it's going to get very concentrated down to the 1200 milliosmoles. As it comes through here, there are no aquaporins, right? So there are no AQP channels, so no aquaporins. And again, an aquaporin is literally a water channel that allows one water molecule at a time to go through that channel. And initially, the ions, because it's concentrated here, are just going to be able to are going to go through their just passive diffusion. But as it gets up here, we'll have to go against this concentration gradient. Well, initially, remember, this line represents cells, and so that's why I drew this out, so you can see this specific cell. And of course, inside of the cell, as you know, the inside of the cell doesn't have high sodium levels. It's high out here and high out here, but it's not high inside of the cell itself. And so the sodium is going to enter in, following its concentration gradient, and it's going to bring in it with a potassium and 2 chloride ions. So that's the sodium, potassium, 2 chloride channel. And now you want to get the sodium to really go against its concentration gradient. So you just use typical sodium potassium ATPases, and then the potassium is recycled because potassium is pretty high in the cell, but it can come out over here. And this is a ROM-K channel. Let's put ROM-K. So, and the potassium here is going to help with the reabsorption of the calcium. And oh, this arrow's in the wrong place. We're going this way, right? We're bringing in, we're taking the calcium, magnesium that's here, magnesium 2+, and we're bringing it out. We're being reabsorbed because remember, this is the blood. Okay, this is the blood supply. So anything that comes out, we're going to reabsorb it. And this actually happens through a paracellular pathway, paracellular transport. And this you absorb most of the magnesium, like 60 something percent of the magnesium and like 25% of the calcium through this way. So it's a very important way. But again, these are the main mechanisms I want you to consider. Is on the apical side, this is where the lumen is at. Okay. You have the sodium, potassium, 2 chloride channel, and it's bringing those into the cell. And then you have on the basolateral side, right? That's where the blood, I think a basolateral for B for blood, this is going to be reabsorbed. Okay, you have the sodium potassium ATPase, and now you can bring the sodium out into here, and this is what's going to be concentrating. So you're going against your concentration gradient. And so this first one here, of course, this is secondary active transport because your sodium is following its concentration gradient because you are bringing that sodium into the cell, following its concentration gradient, and establishing the gradient with the sodium potassium ATPase, which is a primary active transport because you're directly using ATP. Now, we're able to get a lot of the sodium out. So this is going to absorb another 20% or so of the sodium. And over here, when you talk about 67%, so 20%, 77, 87% of the sodium is almost all the way reabsorbed. And now we're going to start fine-tuning things and start reabsorbing the remaining of the sodium through the distal convoluted tubule and even in the collecting duct right here, the collecting duct. Okay, so again, we want to make sure we understand there are no aquaporins right here. And that's going to make it so that as you start pumping ions out of the water, or out of this lumen, what's going to happen? The water stays in here, and you pump the ions out, so the gradient is going to start decreasing, the osmolarity. So here it's like 800, and over here it's 300, and then 200, 100, right? Because you're getting rid of all those ions, and water can't follow it. But over here, because you're pumping all these ions out, well, ions can't get through the membrane, so you can't have any passive diffusion. But water, there are aquaporin 1 channels where water is just freely throwing out. And then again, remember the vasa recta, it would actually be going this other way, right? If we turn the nephron around, it'd be going this way, and it would be carrying all these ions for the reabsorption and everything, and that's how this process would continue. Okay, so now we've talked about this ascending limb of the loop of Henle. Now we want to go to the distal convoluted tubule, okay, over here. And we're going to reabsorb the remaining of these electrolytes. So the first thing you have is a channel right here, and you're going to get this your sodium chloride, okay? And pretty much this is similar to this is this sodium chloride channel. Sodium and chloride are going to come in through following the concentration gradient, and then you have over here another sodium potassium ATPase. Okay, so just your sodium potassium ATPase to go onto the basolateral side, right? So here of the cell. Again, you have the apical membrane, sodium chloride transfers through there, and then you have the sodium potassium ATPase pumps, and that's going to get the rest of the sodium into the bloodstream, into the basolateral side, where your interstitium is at, so you can reabsorb everything. So that's going to be another major part of your to be able to reabsorb most of your sodium. Okay, so we have the sodium being reabsorbed over here, and then over here there's actually another cell type. We call it these principal cells, basically. And so we'll just say PC for principal cells. These ones actually respond to this hormone aldosterone. Okay, and aldosterone, what it's going to do is it's going to increase a specific transporter that's specific to sodium, so you can reabsorb even more of the sodium. All right, so let's make this simple here. All right, so we're increasing the sodium levels through the action of aldosterone on these principal cells, and you also have them over here as well on the collecting duct. Okay, you have more of these principal cells. So you're doing the same thing. You're gonna want to absorb more of the sodium. Now, because what's happening? There's two things actually that are happening when it comes to this. Aldosterone is a hormone, and so it's going to bind to its basically it's actually the transcription part is going to go into the DNA basically and cause the production of more proteins. What are those proteins? Well, I talked about how you need to have the sodium potassium ATPase pumps or at the basolateral side. So this is going to end up increasing your ATPase pumps. Okay, so that's going to be one thing it's going to do, and it's going to insert this other channel, just this sodium channel right here on the apical side of the membrane. So you get more sodium that's leaving the collecting duct going into the principal cell. So you literally have more of these channels that are created because the aldosterone hormone created that, as well as more of the sodium potassium ATPases that are going on. And this is, again, occurring in the principal cells. So that's a powerful that's a powerful mechanism of aldosterone, and that's going to create water to be more concentrated upon the action of another hormone called vasopressin. So these same cells also have, I'm just going to draw them out like this as well. They have they have a receptor for vasopressin, and let's say that. So we'll put another name for vasopressin. It's called antidiuretic hormone, okay, or vasopressin. Vasopressin. And this was going to bind to this receptor, like this V2 receptor. So it binds to this V2 receptor, and this is actually a G protein coupled receptor. So it's going to do your typical thing, right? You increase your adenylate kinase, you make your more CAAMP, activate PKA, and then PKA is going to do this very important thing. So you have these vesicles over here, and these vesicles contain another aquaporin. All right, so right here, these are called your aquaporin 2 channels. There's like a lot of different aquaporins, like 10 or more different aquaporins, but aquaporin 2 is basically sensitive to the effects of vasopressin. And what happens is these aquaporin channels, when they get activated like this, they're going to come and do like exocytosis, and they're going to come and insert themselves right here at the apical side of the membrane. And when that happens, your water molecules are going to be able to come through these channels. And once they're there, it's perfect because there's already aquaporin channels right here. There's already aquaporin channels here, like 3 and 4. Okay, so it's aquaporins 3 and aquaporins 4 channels on the basolateral side, but that's not going to help anything if the water can't get through here, right? This side here is normally impermeable to water, but we can insert aquaporin 2 channels via the action of ADH, vasopressin, and then once the vesicles fuse with the membrane, then the aquaporin 2 channels are able to bring the water molecules into these principal cells, and then the water can flow out, and it's just going to follow the sodium levels that from the action of aldosterone, right? So aldosterone is going to the DNA and putting more of the sodium channels in, making more sodium potassium ATPase pumps. Now sodium is going to be higher over here, and then of course water can follow that if vasopressin is available, so we can absorb more of that water. And so this is another critical thing. Vasopressin has so many very important effects, but I'll just add one interesting note is, with children, sometimes they'll wet the bed, and one of the potential reasons is because they don't have as high levels of antidiuretic hormone or vasopressin at nighttime. And so what happens then is the water, right, that's being put all through here only gets concentrated so high, right, 100, 200 milliosmoles or whatever, and it's going to come through here, and there's no vasopressin, so the water, it can't get out of the collecting duct, and so if it doesn't get out, it's going to just be excreted into the urine. So then the bladder just gets filled up and then spontaneously releases its contents. But if you higher levels of vasopressin, what's that going to do, right? It's going to create or cause these vesicles to be put into the membrane, fuse there, and then the aquaporin 2 channels are there, water can flow through, and then water can come out into this higher concentration, right? Because remember, this is very high concentration area, so water obviously wants to come out. And of course, you have aldosterone going on too. It's going to be potentially even more concentrated. So this walks us through a very important pathway of how our nephrons are concentrating urine, as well as how it regulates our blood pressure, actually, because blood pressure is a function of our cardiac output and our volume, right? Our blood volume. So if we decrease our blood volume because maybe we were dehydrated, then our blood pressure is going to go down, and that could be potentially problematic. And this is an elegant process of which we can concentrate our urine and absorb more water or not. And as I said, because this can go down to 1200, and then if you have these different channels over here absorbing more water, so the range is pretty large. It's going to be around 50 to, say, 1200 milliosmoles, osmolarity. And so, again, just think about what's going on, right? If we want to preserve our water molecules, then we want to have very concentrated urine. Because we want to absorb as much water as possible. But if we're drinking a whole bunch of water and there's no reason to conserve that, then we actually want to reabsorb as much sodium as we can, and we want to we're going to have urinating a very diluted, right? So around 50 milliosmoles osmolarity of water. I say osmolarity, there's also osmolality, and that's kind of more what we're doing. Osmolarity is osmoles per liter, but osmolality is osmoles per kilogram. And It's probably more accurate, but in normal, but relatively dilute solutions, they're fairly interchangeable. Okay, let's talk more though about how we're regulating our blood pressure and our sodium levels, because there's a very important system called the RAAS system, we can call it. We'll just do RAAS. That's the renin-angiotensin-aldosterone system. And what happens is, let's start. You're over here over here with the afferent arteriole, and that's all your plasma proteins. Well, there's one important plasma protein called angiotensinogen. All right, so angiotensinogen. And this is made by the liver. Okay, and it's always in the plasma. And this is not active at all. That's the angiotensinogen. But what can happen is it can get converted to something more active, which is called angiotensin 2, or sorry, angiotensin 1. And we'll just abbreviate that. So angiotensin 1. So as angiotensinogen, angiotensin 1. Well, how does that happen? What converts this? This is done by this enzyme called renin. Well, where do we get renin from? Renin comes from right over here. So here's renin is actually being produced from these cells right here. Okay, this makes the renin molecule, and these JG cells, the juxtaglomerular cells, or the granular cells, they produce this enzyme renin, and renin essentially activates angiotensinogen to angiotensin 1. And now angiotensin 1 doesn't do a whole lot. Instead, well, right here, we'll take angiotensin 1 and we're going to convert that to angiotensin 2. Now, angiotensin 2 is what does everything. But what does this part? This is going to be done by ACE, angiotensin converting enzyme. And this is found in epithelial tissues. The lungs is often given as a prime example of that. We know, like with COVID, for example, the SARS-CoV virus attacked the lungs because of a lot of these receptors in there in the epithelial cells. But this is the angiotensin converting enzyme. So you get angiotensin 1 that's going throughout the body, gets converted to angiotensin 2. And then what does angiotensin 2, what does that do? Well, actually, we can make sense of it if we understand why renin was produced in the first place. Well, renin was produced because you have this macula densa right here, and the macula densa is actually sensing changes in sodium chloride levels. So I can just put delta sodium chloride levels. And it's very sensitive. And so as it senses different changes, it signals through a certain mechanism to the JG cells, and that produces the renin, and then the renin causes all this to happen. And angiotensin 2 then, what does that do? Well, let's look at a couple of things. One of the big ones is this going to cause thirst. So it goes to the hypothalamus and causes thirst. It also causes the release of ADH. Where do we hear ADH before? That's right, vasopressin, right? So the hypothalamus is also going to release ADH in response to this process. It's also a very powerful vasoconstrictor. Vasoconstrictor. And because if we think about what's happening, all right, so let's say that you have a loss of blood volume, okay, your blood flow has decreased. Well, how are you going to sense that? If you have a lower blood flow because you're dehydrated, if you have lower blood flow, we can write this down. So let's say you have dehydration. So dehydration is going to cause a lower blood volume. if you have a lower blood volume, that's going to cause a lower what? A lower blood pressure, right? And so you have two things going on. So think about what's happened here in the juxtaglomerular apparatus. You have lower blood pressure, so you have lower hydrostatic pressure, all right? And so you have less filtration that could happen. Now, this is actually a big deal because, as I said, we want to filter about 125 milliliters per minute. And this has to all change because if you're changing your blood pressure from whatever problems happen, like dehydration, heavy exercise, or whatever it is, you don't want to have a different glomerular filtration rate. You want to keep that very tight. And there's several ways that we can do this. And I should have talked about this earlier, but you can change the afferent arterial, the diameter, vasoconstriction or vasodilation, or the efferent arterial, the diameter. So if we think about what's happening, if we do vasodilation of the afferent arterial, so the blood's coming in, we open that up wider, then you have more blood that's going to be coming in here. If you have more blood, then it's going to increase the pressure. If you increase the pressure, right, then according to our equation right here, they're going to increase the net filtration. Another way to increase the pressure is you could constrict the efferent arterial, right? If you constrict the efferent arterial, then the blood comes in and then it can't go out quite as easily. So that can also build up pressure inside and you can get that filtration. And then the opposite is also true, right? If you do a constriction of the afferent arterial, then not as much blood is going through, and so you have a lower hydrostatic pressure, so you have lower filtration. Or you could do vasodilation over here, and so you like open up the drain more, and there's going to be less hydrostatic pressure, so you get less filtration. So if you have changes in your blood pressure via blood volume or whatever is going on, then the efferent and afferent arterial can constrict and dilate, and there's a number of other things going on as well that can help with that. In fact, there's like with some of these capillaries, some of these can change. You have different changes in the surface area as well, so that you can maintain your glomerular filtration rate, so it's really quite stable. Now, as I was saying though, if you are dehydrated and you have low blood volume, so therefore you have lower blood pressure, and you're going through this afferent arterial, well, how are you actually sensing that? Well, in this case, you have your macula densa that's going to sense like, hey, I'm getting less sodium that's being taken up, so there must be an issue here. So the macula densa then tells the juxtaglomerular cells, hey, we need to make some renin. So they make renin. It comes over here, does this pathway. Finally, you get angiotensin 2. And then this is going to stimulate you to say, hey, I am thirsty. I want to get some water. It's going to also increase ADH or vasopressin. And what's that going to do? It's going to come over here and put the aquaporin 2 channels in the collecting duct. There's actually some that's going on over here as well, but both aldosterone and vasopressin act, both these areas. But then you're going to be able to absorb more water into your blood, and you make your urine more concentrated. Right? You also have the vasoconstriction that's going on, because if you can constrict all of your blood vessels, then that's going to increase your, it's going to allow you to have a higher cardiac output so you can maintain your blood pressure, right? So this angiotensin 2 is a very powerful molecule. It's one of the stronger vasoconstrictors, actually. And it's all sensed, a lot of it's sensed just right here with the macula densa. It's very, sensitive to these changes, okay? Okay, so that's our basic renal physiology. Let's just walk through this one more time for a quick recap, all right? Here we have your nephron that's going to filter your blood. The filtration starts here where you have your peritubular capillaries, and you have your hydrostatic pressure that's pushing the blood into this area. You're going to filter at 125 milliliters per minute to get to 180 liters per day. Very impressive. And most of everything will be absorbed right here in the proximal convoluted tubule. 67% of your water and sodium chloride will be reabsorbed right there. And a lot of your bicarbonate is, and pretty much all of your glucose and amino acids. Unless you hit the saturation point for your glucose, then you get like diabetes, which that's why you end up getting glucose in your urine. Now we want to concentrate this because we don't want to, if you didn't do this pathway that we're going to talk about, then you would have to urinate also 180 liters per day. And no one wants to do that, or you could not even drink that much water. So this is why this is very important, because you have in the descending limb of the loop of Henle, this has a lot of aquaporin 1 channels, so water can go through, but your ions cannot go through, so it's impermeable to the ions. And as you go down deeper, you reach the bottom of the loop of Henle and deep into the medulla, because it's a juxtamedullary nephron. You reach down to 1,200, maybe even 1,400 milliosmoles. As you come back up, this is your ions are able to go through passive diffusion, and like your sodium and your chloride, it just follow this concentration gradient. But water can't go through because there are no aquaporins in this area. And as you get to the thick portion of the ascending limb, you have these cells that are literally pushing or pumping out the ions against this concentration gradient. Well, the first on the apical side, you have the sodium potassium 2 chloride transporter that actually is going down its concentration gradient because you don't have high sodium levels inside the cell. But then to pump it out into this area, you use the sodium potassium ATPase pumps. You get a lot of sodium outside. You can absorb another 20% of that because, again, this is all blood. You have the vasa recta. That's all right here, bringing things all the way around, right? Getting all the ions and the water and so on. And you also have the potassium. Then once you pump it back in, it can go through. It kind of cycles through these ROMK channels, and that's also important. So you can reabsorb the calcium and magnesium through some paracellular transportation, right? And as you do that, though, you can really lower the osmolarity a lot, down to 200, even 100 milliosmoles. And again, this portion here is actually the same portion over here, because remember, it's twisted upon itself. And now you get to the distal convoluted tubule, and you have your sodium chloride channels, again, going with its concentration gradient, and then you have the sodium potassium ATPase, so you're reabsorbing basically the remaining sodium. Then you have the principal cells over here, and on the latter portion of the distal collecting duct of the convoluted tubule, and then you have on the collecting duct right here, the principal tubules. And these are sensitive to aldosterone, and aldosterone is going to help you to reabsorb a lot of the sodium because it's a hormone. It's going to cause the DNA to create more of the specific sodium channels on the apical side of the membrane, as well as get more sodium potassium ATPase pumps, so you can absorb more of that sodium. And then if vasopressin is present, that's going to come and bind to the V2 receptors, the G-protein coupled receptors, so you get the pathway of the denolite kinase making more CMP, activate PKA. Then PKA is going to go to these vesicles here and insert more aquaporin 2 channels into the apical side of the collecting duct. Water is able to flow through the aquaporin 2 channels and then lead to the aquaporin 3 and aquaporin 4 channels. And then we can reabsorb that water if needed. And therefore, you can get your urine to be concentrated up to 1,200 milliosmoles. Of course, there's no aldosterone or vasopressin specifically, then you could have urine as diluted, say, 50 milliosmoles per liter. So this is a typical quick way of how our body is able to maintain our hydration status, maintain our blood pressure, and maintain our glomerular filtration rate around 125 milliliters per minute. And it's all being sensed by this macula densa, because again, if there are problems like changes in the amount of sodium chloride, we're saying, hey, there's not enough, something's going on here, then what's going to happen? The macula densa will sense that, tell the granular cells, the juxtaglomerular cells, to secrete more renin. Renin will then convert angiotensinogen produced by the liver to angiotensin-1. Then angiotensin-1 will be converted via the epithelial cells, which is concentrated in the lungs, via the angiotensin converting enzyme to angiotensin-2. Angiotensin-2 will go to the hypothalamus to stimulate your thirst and also your antidiuretic hormone or vasopressin right here, as well as actually a vasoconstrictor, so we can get that on the arterioles and protect our blood volume, and as well as maintain our cardiac output to maintain our blood pressure, right? And now there's other really important things to talk about with the kidney. Also, is really important to maintain our acid-base balance. So I'm going to redraw some of these sections, and then we can focus and zoom in on how the kidneys play in regulating our acid-base homeostasis.